Okay, well, we're gonna give this a go. So here's the deal. I don't quite know exactly how to do a video like this, but we're gonna give this a go. And so this is when you can point up here to baby boomer and say, okay, boomer, we're gonna give it a go. So with that in mind, I'm gonna close this so I don't get distracted by me. All right. So think back to the central dogma of biology. We talked about DNA to RNA to protein. Through transcription, we've made the first step, DNA to RNA. The information that's contained in DNA has been transcribed or copied into mRNA, and this disposable, expendable copy of the genetic information is now ready for processing. So next, we're going to follow that copy into the cytoplasm and translate that information into the language of proteins using amino acids as the words. A question I might throw to you, though, is this. Can you explain what happens to that initial transcript, the pre-mRNA, before it can be exported? Remember, the pre-mRNAs have to be edited. The ends of the molecule have to be protected before the journey to the cytoplasm can occur. So can you describe those edits? And can you describe the enhancements to the molecules? I certainly hope you can. So, moving on. All right, on the last slide, I mentioned leaving the cytoplasm. Keep in mind, in prokaryotes, all of this occurs in the cytoplasm. As we move forward, I'll try to remember to highlight any procedural or functional differences between eukaryotes and prokaryotes. So what is translation? Simply, the cell is going to read the mRNA message and assemble amino acids to create a polypeptide. Now, from an earlier unit, remember, a polypeptide chain is the primary structure of a given protein. It is a chain of amino acids specific to that protein. A polypeptide chain is not the same thing as a protein. A functional protein is not just a polypeptide chain, but typically multiple polypeptide chains that have been precisely twisted, folded, coiled, and otherwise manipulated into a molecule that has a unique shape. And that shape determines how the protein works. Now, back to translation. We have mRNA. We'll now include tRNA, transfer RNA, to our protein party. That's right, I said protein party. We need a site for the party. That's going to be the ribosome. Once we get all the participants gathered at the ribosome, a highly orchestrated dance is going to proceed. So let's take a look. So the translator in this system is this guy right here. This is tRNA, okay? The function of tRNA is to transfer amino acids from the pool of amino acids that are found in the cytoplasm to a growing polypeptide chain. The key to this process is this simple fact. Each tRNA molecule must translate a given mRNA codon into a certain amino acid. This is possible because the tRNA has a specific amino acid at one end of the molecule, while at the other end is a nucleotide triplet that can base pair with the complementary codon on the mRNA. This accurate translation of the genetic information relies on two instances of molecular recognition. And the first one's taking place in these places, okay? This is a correct matching up of the tRNA. Oh, look at that. There we go. Correct matching up of the tRNA and amino acid. This is shown on the slide and is carried out by a set of enzymes called aminoacyl tRNA synthetases. The second molecular recognition is carried out on or in the ribosome, pairing the tRNA anticodon with the appropriate mRNA codon. There. Sorry, I kept the spotlight on too long. All right, so the tRNA molecule consists of a single-stranded RNA that's about 80 nucleotides long. This slide shows, right here, this shows on the first image on the left, the two-dimensional structure of the molecule. An amino acid binding site or attachment site is shown here at the 3' end of the molecule. Note this site always ends in CCA. CCA. The anticodon triplet is at the opposite end, down here. This portion will base pair with a specific mRNA codon. The other images are just different structural views. Make sure you do notice that the structure is folded and held together by hydrogen bonds, following the base pair rules for RNA. The tRNA molecule is the translator. It's going to read a nucleic acid word, the mRNA codon, and it's going to interpret it as a protein word, the amino acid. In eukaryotic cells, the tRNA is made in the nucleus, then travels to the cytoplasm. In both prokaryotes and eukaryotes, the tRNA are used over and over. So let's talk about wobbling. The codon-anticodon relationship is, like many relationships, complicated but flexible. If one tRNA variety existed for each mRNA codon specifying an amino acid, there would be 61 different tRNAs, but there's only 45. So what does this mean? Some tRNAs must be able to bind to more than one codon. And this is possible because the rules for base pairing between the third nucleotide base of a codon and the corresponding base of a tRNA anticodon are relaxed compared to those at the other codon positions. Example, a nucleotide base U at the 5' end of a tRNA codon can pair with either an A or a G in the third position at the 3' end of an mRNA codon. Flexible base pairing is called wobble. Look at the codon charts you have, and you'll notice that you'll see differences at the third position, but not at the other base positions for similar codons. We're almost there. All right, ribosomes. So where do we have the translation party? Well, we have that at Club Ribosome. The ribosomes are going to bring together our participants, sort of like an 8th grade dance. We have the tRNA participants and the mRNA participants. They know they want to dance, but they don't quite know how to pull it off. The ribosome is the matchmaker. The ribosome is made of 2 protein subunits. In eukaryotes, these subunits are created in the nucleolus. Following typical protein protocol, there is a transcription and some imported help from the cytoplasm in the nucleus to complete the subunits. The subunits then get shipped out of the cytoplasm or to the cytoplasm. Here we've got a ribosome, and you can see it's made up of a large and a small subunit. They form a functional ribosome only when they're attached to an mRNA molecule. So here we have the anatomy of a functioning ribosome. I guess our dance party analogy has just added 2 more participants. Is this now country line dancing or the electric slide? Okay, boomer. Here in this image, you can see all of the important bits of the functioning ribosomes. A nice exit for the peptide chain and docking sites, which I'll cover in the next slide. Here is a schematic model showing binding sites. The ribosomes have binding sites that help bring together our dance participants. There's a binding site for mRNA and 3 binding sites for tRNA. The P site holds the tRNA that's carrying the growing peptide chain. The A site holds the tRNA for the next amino acid to be added to the chain. And the E site, the exit site, that's the finished tRNA will leave the ribosome from the exit site. So make sure you're clear on what each of those does. Here are all of the sites occupied and doing their individual thing. Evidence in current research leads us to believe this coordination of structure and function are a result of the rRNA that makes up a majority of the ribosomes and not the proteins. I think that's twice in recent days we've downplayed the role of proteins, which I build up so much early on. And so what I want you to do is tune in next time for live action translation. Woohoo! Live action.